Well, thank you. As introduced, I'm Tony Pattison and I work for the Queensland Department of Agriculture and Fisheries based at South Johnston Research Facility, North Queensland. And the work I'm presenting is a collaboration from many people who have been involved in research projects to manage tropical race 4 in Australia. Now I'm focusing this presentation on the work we have been doing with non-banana plants and the role they play in Fusarium wilt management. Firstly, I'd like to give some context of why we are pursuing this avenue of research and what is already known. Next, I'd like to focus on the alternative host, providing some results from glasshouse, field and laboratory experiments. Then I'd like to discuss the role of non-banana plants as potential rotation crops with results from glass and field experiments before moving on to their role as ground covers. At the conclusion of the presentation, I hope that you'll find that non-banana plants do have a role in their management of Fusarium wilt and can be either desirable, that is, reduce the disease, or undesirable and sustain banana Fusarium wilt. And as we have already heard throughout this series of presentations, TR4, tropical race 4, is spreading globally, causing devastations to banana plantations and livelihoods. However, in Southeast Asia, banana growers and researchers have been dealing with the disease for more than 20 years and have developed some quite innovative banana production systems. But banana growers face three scenarios regarding the disease. Firstly, they may not have the pathogen and they need to keep their farms disease-free, but can largely continue to focus on production. Secondly, the disease appears for the first time and the focus becomes on limiting the spread internally and to keep the farm as productive for as long as possible. Finally, the disease can be overwhelming and growers need to look at how they reduce their losses and start again. In each of these scenarios, the management of Fusarium wilt changes. And when the farm is free from disease, the emphasis should remain on biosecurity and doing everything as possible to remain disease-free. However, as this is a long-lived soil-borne disease, there are numerous pathways onto the farm, and the end point is when growers need to consider alternative cultivars like this photo, GCT-CV218, which is being used in the Philippines. However, following the first incursion of the pathogen, farm management practices become crucial. Containment largely focuses on destruction of infected plants, either through burning or urea fumigation, but other practices should be considered. And to show this in another way and highlight some of the options of different management strategies, on the left of the screen, we have biosecurity practices like restricting farm access, zoning, fencing, clean planting materials. On the far right of the screen, we have developed some of the resistant cultivars through some clonal variants, hybrids, genetic modification, and gene editing like we heard last week. What I'm focusing this presentation on is mostly during that management phase and the role non-bananas play in management of the disease. There are other management options that we've just heard about for growers that have touched like in managing inputs, biocontrols, and crop destruction. So just firstly moving on to the banana production in Australia. There are different VCGs of Fusarium in the different banana producing locations, as Stuart Lindsay pointed out in the first series of presentations. Each production has its own unique geographical features. Our main production area is in the northeast of the country, in the humid tropical area, which is relatively warm and wet year-round, producing around 95% of our bananas. Then there is a subtropical area that is much cooler during the winter months and has lower rainfall, producing around 4% of the production. And thirdly, we'll be focusing on a small production area near Darwin, which has higher uniform temperatures and a very monsoonal rainfall pattern. In each of these banana production areas, though, the cycle is similar. So excluding quarantine restrictions, if we start with an infected crop of Fusarium wilt that has been devastated, that crop is destroyed, the land is fallowed, we replant bananas, And what we are hoping for, that we are successful at reducing the pathogen when we replant and allowing as many returns as possible so we can market bananas. What we also hope is that over the same period of time, we increase antagonistic organisms to help suppress the pathogen. Non-banana plants can have 3 roles in this production cycle. They may carry Fusarium through either to the next crop as alternative hosts. We also need to consider how to manage the fallow period and what to grow. And finally, we need to consider non-banana plants as ground covers, where we can manipulate and select what grows in amongst the plants to manipulate their alternative host status and encourage antagonists. And I'll go through these in more detail shortly. So what we also know from the work of Nolan Warman and Liz Aitken and many others about the infection process of bananas and how it moves into the roots and through the xylem tissue with abundance of chlamydia spores being produced once the plant senesces. We also know from the work that Paul just presented that he and his PhD student Henry Burt did, what happens when we remove the microbiome from the soil. As Paul was saying, they added FOC to sterilise soil and fresh soil and measured its abundance 21 days later and found a small increase in the fresh soil, but a 2,500-fold increase of FOC DNA copies in that sterile soil. And if we repeat the same process but use susceptible plants, as my colleague Hazel Garza did in a pot experiment, we get the same result. The disease is much more severe in the sterile soil than in the fresh soil, indicating that other soil microorganisms actively compete with Fusarium, reducing its dominance. We also know that over the last 100 years, there have been around 938 publications on banana Fusarium, and this has increased dramatically in the last 20 years as tropical race 4 has become a global problem. Most of these publications come out of China, India, USA, and Australia. However, only 3% of these publications have anything to do with non-banana plants as either alternative hosts, rotation crops, or ground covers or intercrops. Therefore, there is a large gap in our knowledge about these plants in the banana production system that needs to be addressed. So firstly, let's consider non-banana plants as alternative hosts in their role in FOC inoculum management. As part of this work, as part of a project, the field surveys were conducted by many people in different banana producing areas, starting in the subtropics where race 1 and subtropical race 4 dominate, then in the humid tropics where race 1 is dominant, and then finally in the Northern Territory where TR4 is dominant. The survey in the subtropics collected 46 species and found race 1 on 6 of them and subtropical race 4 on 1. In the survey in the humid tropics, 115 samples were collected from 20 different plant species, with race 1 recovered from 4 species. In the Northern Territory, TR4 was recovered from 6 alternative hosts by Charles Mintoff and his team up there. However, in glasshouse experiments from plants recovered from northern New South Wales, only 8 plants were tested, where 6 plants were found to be hosts to race 1 and 1 to subtropical race 4. A similar glasshouse experiment using plants found in North Queensland tested 50 potential hosts against subtropical race 4, and it was recovered from all species. In the Northern Territory, 12 common weed hosts were tested and a similar result was obtained, where tropical race 4 was recovered from all species. And in a laboratory study conducted at the University of Queensland, Jay Anderson, who's part of Liz Aitken's team there, investigated the colonisation of 3 alternative hosts using a GFP-modified subtropical race 4 isolate. 2 plants tested are common weeds found in banana plantations. Jay found limited colonisation of the roots by the fungus, but found occasional hyphal nets, chlamydia spores, and colonisation of epidermal cells. However, what was really interesting was when Jay treated the plants with herbicides and killed them, the GFP FOC was able to extensively colonize the dead weeds with mass sporulation occurring. So from the work that has been done on alternative hosts in Australia, a grower's guide has been produced. The guide gives some basic information like name, location, description, and the rating based on glasshouse results and whether it has been isolated from field samples. The aim is to give growers some idea of the host potential of the different weeds and whether they are likely to carry over the disease to the next crop. So to summarize the role of non-banana plants as alternative hosts, all plants can be considered as potential hosts to FOC, and the host status really depends on inoculum level, but some plants could be considered better hosts than others. We found it very difficult to isolate FOC from alternative hosts in the field. And the other thing to keep in mind is that when they senesce, such as sprayed with herbicides, they can be extensively colonized by FOC. So now let's move on and consider the role of non-banana plants as rotation crops, which potentially can reduce inoculum and increase soil antagonistic organisms. Firstly, in a glasshouse assay using a method we developed with Wayne O'Neill, we call the pot in pots experiment. This is where we put a small pot inside a larger pot, fill the space between the two with soil and plant our rotation crop. This is allowed to grow for 2 weeks before being inoculated with FOC. And the rotation crops then grow for a further 12 weeks before being harvested. At harvest, 20% of the tops up to 50 grams are returned to the soil, the inner pot removed, and a susceptible banana plant grown in the center. And then these are compared to our controls, an inoculated and an uninoculated bare soil. And then disease assessments, the plants are grown for a further 12 weeks, the banana plants, before being harvested and assessed for disease, including re-isolation from the roots. So if we compare the recovery of subtropical race 4 to our inoculated and uninoculated banana controls, there was a range of responses. Some plants, like peanuts and brassicas, appear to be very good hosts to FOC. There are many intermediate hosts, but we found 4 species that had similar recovery to FOC to the uninoculated banana control. And these are Dallas grass, Queensland blue couch, Burgundy bean, and Leucaena. And these are worthy of further investigation. In a field experiment conducted by Charles Mintoff and his team in the Northern Territory, which he alluded to last week in his presentation, several rotation crops for the region were tested. I'm only going to present the results from 4 of the treatments: a weedy fallow, which was our control, cavalcade, a Centricina pascorum, jarrah-grass, Digitaria meligiana, and sorghum and sugar graze of forage sorghum. Charles used a method developed in a previous project to measure the FOC TR4 DNA based on a TACMAN assay. And the samples corresponded to the previous crop of infected bananas, the rotation period when the different crops were grown, and the subsequent banana crop following the rotations. And as you can see, there was very little difference, and it certainly was not significant in the TR4 DNA recovered from the soil between the different treatments or between the different phases in the crop cycle. However, there were differences in disease incidence determined by the external expression of Fusarium in the subsequent banana crop. If we use our weedy fallow as a reference, there was a rapid increase in symptoms 21 weeks after planting, reaching around 40% of infected plants at the plant crop. Cavalcade, the Centricina species, was significantly slower to express the symptoms and had a 20% expression of disease symptoms by harvest. The jarrah-grass, on the other hand, was much more rapid and reached 50%, and the sugar graze intermediate. So while rotation crops didn't reduce the pathogen in the soil, as all treatments were equally infected, the cavalcade did slow the expression of the disease symptoms. So I'd now like to move on to ground covers and the role they may play in the management of banana Fusarium wilt. From some earlier work which I published with Emily Rames, we found that when vegetative ground cover was maintained around the banana plants, there was a reduction in the amount of necrotic tissue within the banana pseudostem. Although all plants were still infected, necrosis was significantly reduced over 3 crop cycles. Associated with this reduction in pseudostem discoloration was an increase in the fungal richness under the vegetated ground cover over that same period. And fungal richness was also found to be associated with activity of the enzyme beta-glucosidase, so that an increase in fungal richness was highly correlated to an increase in beta-glucosidase. And both an increase in beta-glucosidase and fungal richness were correlated with a decrease in the pseudostem discoloration on a 1 to 6 scale. I tried to repeat this finding in the Philippines, working with our partners from the University of Southeastern Philippines, where we had 2 field experiments on tropical race 4 infected land. In the first case, at Lasang, our ground cover treatments in the green bars failed to alter the beta-glucosidase activity relative to the bare soil in the red, with the red bars. Similarly, at the second trial site at Kapalong, a vegetated ground cover treatment failed to alter beta-glucosidase activity. Therefore, it wasn't surprising that the incidence of Fusarium wilt was not significant between our ground cover treatments, the green and the red bars, regardless if we use susceptible banana cultivars like Grey and Nain or a more resistant cultivar like GCTCV 218. Again, we had a similar result at our second trial site at Kapalong, which had a much higher inoculum level and a higher incidence of Fusarium of tropical race 4 infection. Although there were differences due to cultivars used there, I believe there were several factors that were really important in the experiments to why we did not see any effect due to ground covers relative to the bare soil. The primary reason was the overlying management that was imposed, as both experiments were conducted on commercial banana farms where ground covers were not the normal practice. Despite our best efforts to establish and maintain the ground covers, the contract workers that were employed there were doing their best to remove them, and then we were applying overriding farm management practices such as high nutrient inputs. And the second reason we didn't see any differences was time, as time is required to increase the antagonistic organisms when management practices change. It is not instant. And as Paul showed with his slide earlier on, we have some preliminary results from a second field experiment conducted in North Queensland, which used Illumina next generation sequencing to track changes over a 4-year period. In this case, we used a number of Fusarium oxysporum OTUs as a surrogate for FOC because of the quarantine restrictions on working with Fusarium wilt in Queensland. Following the establishment of the treatments, the bare and the vegetated, there was an increase in Fusarium oxysporums in the bare soil, the silver line, compared to the vegetated ground covers, the yellow line. At around the 36-month, 3-year period, there was a significant increase in the abundance of Fusarium oxysporums OTUs in the bare soil, which then declined back down to at 42 months. However, throughout the trial, the number of Fusarium oxysporum OTUs always remained above that vegetated ground cover treatment until the termination of the experiment. But at the 31-month mark, what was really interesting was that there was some really unfavorable climatic conditions. First, really heavy saturating rainfall, followed a few weeks later by extremely hot, dry conditions. And we'll be investigating further whether the impact of climatic extremes helps this dominance of Fusarium oxysporums, and then also the effect of the buffering effect that the vegetation of ground covers may have. So I hope from the presentation that I've given now that you are able to see the different roles that non-banana plants have in management of banana Fusarium wilt at the different stages in the banana cropping cycle, either as alternative hosts, ground covers, or rotation crops. And they can either be positive or negative on the FOC inoculum and with extrapolation with antagonistic organisms. And then another way of what we've been looking at to discuss is through a conceptual structural model. If we have our non-banana plants, they can either be alternative hosts, rotation crops, or ground covers. And in these roles, they can either increase FOC or decrease antagonistic organisms, such as alternative hosts and some rotation crops that can carry the disease through to the next crop, making them undesirable. Or non-banana plants can be grown as rotation crops and ground covers to reduce FOC and increase the antagonistic organisms, making them desirable. The task is determining which plants fit into the different categories, which is something we have started doing for the Australian banana industry. So to conclude, I hope you agree that non-banana plants have an important role in the management of banana Fusarium wilt, but they may be either desirable or undesirable. As alternative hosts, all plants tend to host FOC, Fusarium oxysporum, but some are better plants than others. It is when they are killed, the Fusarium can colonize them as a saprophyte. As rotation crops, some plants aid in reduction of inoculum, which we found in pot experiments, but we are yet to replicate this in the field. What we are seeing in the field appears to be an increase in the tolerance of the plants, which is slowing the external disease suppression. As ground covers, non-banana plants can reduce the dominance of Fusarium, increasing plant tolerance and resilience to banana Fusarium wilt. So finally, I'd like to acknowledge the people who have contributed to this work, and this is just some of them from the meeting held really early in the project. And I apologise for not mentioning everyone's names that contributed, but I'd also like to thank the agencies that they work for, which allowed them to form a really good collaboration, which included state governments and universities. And I'd particularly like to thank the funding agencies that contributed to this work, notably the Queensland Government, Hort Innovation and ACIAR. And so finally, thank you for a chance to present what we've been doing in Australia. And I'll now hand back to the chair.
Thank you, Tony, for your very inspiring presentation. Now I open for discussion. I already seen several questions. So first question is from ACIAR from India, Dr. Savalaya. His question is about, to Tony is, what about a paddy rice for crop rotation? And it has been proved that due to flooding, it can be reduced FOC inoculum?
Yeah, well, what I guess we're looking at is what crops can be grown in an Australian context. So we don't grow paddy rice with bananas in our banana growing area. So it wasn't something that we looked at. I know that there's a lot of other research that is some other research that has been done specifically looking at that. And from what I understand from that literature was that paddy rice was probably one of the more suppressive crop rotation that you could grow with bananas to reduce the Fusarium. What we also know now is from the work that we've been doing with Paul is that the microbiome is extremely important. Where we've got that functioning microbiome, we seem to get greater suppression of the disease. It's when that's lost, and if that gets lost under paddy rice, then it's just a matter of time before it comes back again, before it's reintroduced. So I think that's probably a big avenue for further research, particularly in our area, in our region of Southeast Asia. Sorry, I don't have a straight answer for that one.
Okay, Tony, second question also from ICA, Dr. Longan-san. His question is, how was the yield of banana in ground cover in comparison to non-cover field?
Yep, and that's an excellent question. I didn't show the agronomic data, but I collected a lot of agronomic data over that trial from the second field trial that I presented with the Fusarium OTUs, and the yield was a little bit lower than well, yeah, it was lower under the ground covers. But what we've learned since then is that we have tried to establish the ground covers when we established the bananas. I was running that trial on a commercial farm who kept looking over my shoulder. He's now implemented the system on his farm so that he has 100% ground cover over his farm. He says his yields are actually better because of our plot structure, and I think the early competition from the ground covers when the bananas were getting established. We got a reduction in yield, and the plants never got to catch up. So what we're now trying to do is establish the bananas first, and then reintroduce the ground covers around them once they're up and can handle a bit of competition, so we don't get that that yield penalty.
Okay, Tony, still a lot of questions coming, but I will pick up a last question. The last question, and the other you can answer in the chat. Yeah. The last question is from Gili Babu. His question is about the role of nematodes in Fusarium wilt.
Yeah, well, it's an interesting question, and I was having this discussion today, actually. What I've been finding is, with our collaboration with Paul, I measure the nematode diversity and what nematodes are there, and what the conditions that seem to favour plant parasitic nematodes, where we've got more plant parasitic nematodes, also tend to be the conditions that favour Fusarium. So whether plant parasitic nematodes cause more Fusarium, I'm not sure, but it's those underlying conditions of high nitrogen, lack of diversity, lack of competition, which favour both root pathogens. So it's really hard to separate, are the nematodes causing the problem, or is it the conditions in which they've been selected out on? So but there is a very close association between more plant parasitic nematodes and more Fusarium, less suppressive fields. And to reverse that around, where we get less plant parasitic nematodes and more diversity of nematodes, we get more diversity of other organisms, and they tend to be more suppressive to Fusarium wilt.